Okay, hello. Up to this point, we've in this semester we've been talking about parametric tests. So last week we talked about non-parametric tests, and I'm going to shift your focus again back to talking about parametric tests. But so far, all of the tests we've talked about fall into a category that are referred to as univariate. And by univariate tests, what I mean is there's a single dependent variable that may be affected by one or more independent variables. And we've talked about univariate tests where the independent variables were categorical or discrete. And we talked about univariate tests where the independent variables were continuous. And we talked about where there was a mixture of the two different types when we talked about analysis of covariance. Today what we're going to talk about is multivariate analyses that are, again, still parametric tests, but instead of having just a single dependent variable, we might actually be interested in multiple dependent variables. Okay, so this adds a whole new level of complexity, and I kind of warned you about this when I told you to start charting and making a table to keep straight what kind of test is applied when. In multivariate tests, you may ask the question, how will variable x, my independent factor, affect many different properties of a system or several different properties of a system, in which case I have several dependent variables. So I may have a single independent variable that affects multiple dependent variables. I may have multiple independent variables that affect multiple dependent variables, and finally I may have multiple independent variables which include a mixture of continuous or continuous variables and discrete variables, and that combination of independent variables may affect multiple dependent variables. Okay, so it starts to get a little bit woolly when we start talking about multivariate analyses. So I'm going to introduce you to the two most basic types, or actually three most basic types today. The repeated measures analysis of variance, the multivariate analysis of variance, or MANOVA, and finally we'll talk about MANOVA, which is a multivariate analysis of covariance. Okay, so are you ready? So let's start off by talking about repeat measures and analysis of variance. Sometimes you can imagine experiments such as what I'm showing here. Here we're looking at a plant growth experiment, and I'm interested in this study. We were interested in how effects of different fertilizers, such as municipal biosolids or organic fertilizers, how they affected plant growth. And when we're talking about plant growth, the way we measure that is, we would measure the height or the blade length of these corn plants many times over the course of 3 months. So my dependent variable is plant height, but I have many different measurements of the same plant over a period of time. So effectively what I have then is multiple dependent variables, where on every date that I measure height is my dependent variable, but every day I'm measuring that same property, that same height. So every date has a set of heights that are measured, and then so these multiple replicates, or these replicates over time of plant heights, can be thought of as multiple dependent variables. Okay, multiple measures of the same dependent variable. Okay, so we'll talk about this more in class, and hopefully we can talk about some more concrete examples to help this make sense. So if the property of an individual subject is measured repeatedly over time, the observations aren't independent of one another. So in other words, like if I measure the height of one of these corn plants today, the height today is going to be linked to or affect the height 2 weeks from now. Okay, so a plant that's tall. If I have two plants and one is tall today and one is short today, probably if I come back in 3 days, one that's tall will still be tall and the one that's short will still be short relative to each other. So their heights 3 days from now are linked to their heights today. Okay, so again, the same property measured over and over through time. Each time I measure that measurement is linked to previous measurements. It's dependent on previous measurements. Okay, so here's the example, and I've already talked about it a little bit. I'm looking at plant height. I'm asking the question, does fertilizer treatment affect plant height? I'm going to measure the same plants over and over again through time, and I want to know if there's a pattern in terms of the plant height or the change in plant height over time. So in other words, does the fertilizer affect growth? Okay. When I run a repeated measures analysis of variance, today we're not going to get into the mechanics of the repeat measures ANOVA, but we're just going to talk about it in principle. When I run the repeat measures analysis of variance, I'm going to have three main effects. Okay, there are three basic outcomes that SAS will give me. One is the effect of the treatment. One is the effect of the longitudinal variable, which usually is time, because again I'm measuring that variable over and over through time. And the third thing it'll give me is the effect of the interaction between treatment and time. Okay, so in most cases it's this interaction that we actually care about. So again, if I'm asking, does the treatment affect plant growth? Growth is height, change in height over time, so it's the interaction between the treatment and time that I really care about here. Okay, so let's think about that graphically. Okay, so if I'm measuring plant height as over time as a function of three treatments, a reference treatment, 8 tons per hectare refers to a loading rate for biosolids. 22 tons per hectare refers to a higher loading rate for the solids. What you'll notice is we basically are generating three different linear patterns. Okay, the yellow dots represent one linear pattern, the black dots represent a second linear pattern, and the third represents yet a third linear pattern. Starting at time 0, those dots all overlap with a plant height of 0 when we're looking at the seed stage. So what I'm really interested in then is, with a repeat measures analysis of variance, I'm going to get a treatment effect, and the treatment effect would look like this for my control, my reference. And basically what you can think about is imagine I drew a regression line through my reference. And suppose I took the midpoint or the fulcrum of that line and extrapolated it over to my y-axis. Okay, that would give me the fulcrum for that line, or the midpoint for that line, has a plant height value, corresponds to a plant height value of 29.4. Okay, for 8 tons per hectare, for the 8 ton per hectare treatment, the midpoint of that line intercepts at 36.6, and for the 22 tons per hectare, it intercepts at about 21. Okay, so the point is that the effect treatment is basically telling me where does the midpoint of my regression line intersect the y-axis, and is there a significant difference in that point of intersection with the y-axis. So in other words, a significant treatment effect from SAS output would tell me that 29.4 is significantly greater than 21.2. Okay. Again, is that particularly interesting to us? Not usually. Okay, so it's not usually the treatment effect that we care about. The next one is the longitudinal effect. Okay, the longitudinal effect in this case the time effect, is looking at whether or not there's a linear relationship between my variables, between my values irrespective of treatment and time. So if we were to plot a regression line, and this regression line falls through falls through the black dots here, but it's actually a best fit through all of the points. Okay, this is a fit through all of the points, and we test this, and SAS will test the slope, or when we run the analysis of repeat measures analysis of variance, we're going to get we're going to test for whether that slope is significantly different from 0. If it is, we're going to get a time effect with a p-value less than 0.05. Again, is that particularly meaningful to me? Not really. Not really. It's the treatment by longitudinal effect, or the treatment by time effect in this case, that I really care about. And what a significant treatment by time effect tells me is, in effect, if this slope is significantly different than this slope. Okay? Now, graphically, you look at this and you can say, yeah, I mean, there's a steeper slope for the 8 tons per hectare than for the 22 tons per hectare. But we're going to use the analysis of repeat measures analysis of variance to tell us whether those two slopes are the same, or whether there's a difference in those slopes. And again, if there is a significant difference between those slopes, it's reflected in this treatment by longitudinal effect. Okay, so again, are those 3 slopes different from each other? That's what we're usually interested in, because difference in those slopes tells us that there's a difference in growth rate as a function of the treatment. Okay, sometimes, quite often, when we're working with this type of data or these types of experiments, we'll find that our data are nonlinear in nature. Okay, one of the assumptions of analysis of repeat measures analysis of variance is that our data fit a linear model. Okay, so what would we do in a case like this? If this is what our data look like, what would we do? Well, we'd do a transformation. So we'd transform the data. We'd power transform the data in this case when they plateau. We'd plot it to see whether or not it still looks like it's a linear pattern or a linear relationship. Given that it does look like the data are fairly linear, it's appropriate to run the repeat measures analysis of variance. So here are the slopes that, or the regression lines that fit each of those data series, and we're going to test whether or not there's a significant treatment by time effect. And if there is, that tells us there's a significant effect of the biosolids application rate on growth of the plant. Okay, so I've been slipping up as I'm talking about this, and from time to time saying ANCOVA. So even in my own mind, I'm quite often making this mistake. Repeat measures analysis of variance versus ANCOVA. We talked about ANCOVA before as being a way that we can compare multiple slopes, right? If we wanted to know whether or not there was a difference, like if we think about this example again, we could run an ANCOVA model, and the ANCOVA model basically tells us whether or not these treatments covary with time. Whether or not our data covary with time, and will account for that covariance with time to basically allow us to look at the interaction effect, and the interaction effect tells us whether or not the slopes are different. So up to that point, conceptually, it's very much like what I've just described for repeat measures analysis of variance. So how do you know when to use repeat measures ANOVA versus ANCOVA? The main difference between the two is, let me go back here again. Remember that each of these points represents the same plant measured over and over through time. So this value is dependent on this previous value and forecasts this next value. Okay, so these are not independent measurements, because again, we're measuring the exact same observational unit, the exact same plant, over and over through time. Okay, analysis of covariance assumes that every point that you measure, every single one of these data points that makes up your data series, is independent of any other observation in the data set, right? So if for example we had an infinite number of plants and I measured one plant in this treatment on day 30, on day 40 I measured an entirely different plant within the treatment, day 50 I measured yet another plant in the treatment, then those observations are independent of each other, and analysis of covariance is appropriate. But since it's the exact same plant that I'm measuring over and over and over through time, there's a dependence of one observation on the previous observation. Repeat measures analysis of variances is appropriate then. Okay, so really, again, just in a nutshell, when do I use analysis of covariance? If the observations are independent. When do I use repeat measures analysis of variance? If I'm measuring exactly the same observational unit over and over through time or space. Okay. Okay, the next thing we're going to do really shifts gears, and we're going to start talking about multivariate analysis of variance, or MANOVA, as well as MANOVA, multivariate analysis of covariance. In this case, once again, up to this point we've talked about the effect of a single or multiple factor on one response variable. Okay, what does X do to Y? Now we're going to be interested in the question, what does X do to Y and Z? Okay, so in other words, we're looking at the effect of a single factor or multiple factors on more than one response simultaneously, more than one dependent variable simultaneously. Okay, why would we want to do that? Why would I want to for example, look at the effect of some environmental variable on the Daphnia abundance that we just saw before in this slide, or on the zebra, and at the same time on the zebra mussels that we're seeing on this slide. Okay. Sometimes the decision to use a multivariate analysis of variance depends on the very subtle nature of a factor. So a single factor might nudge a system a little bit. So for example, it may have a subtle effect, but statistically not a significant effect on Daphnia abundance. And at the same time, it might have a subtle but not statistically significant effect on zebra mussel abundance. Okay? But what if that factor, if we considered both, in aggregate, if we considered the effect of that factor on both Daphnia and zebra mussels at the same time, do we now start to see an effect of that factor, right? So in other words, if it increases, if this factor now increases Daphnia abundance a little bit and decreases mussel abundance a little bit, do those two shifts together tell us something about the nature of the factor and how it affects our system? Okay, so again, a single factor may not affect a single variable significantly, but it may nudge a number of different dependent variables and nudge them enough that collectively, in aggregate, we can see that there's an effect of this factor. Okay, so I'm gonna use as an example Great Lakes food web. So these are some of the players in the Great Lakes food web, and a few more players in the Great Lakes food web. But let's imagine this scenario where we've got a lot of different components of the Great Lakes food web. We don't need to spend a lot of time on this on this slide, but we've got a number of different organisms or groups of organisms at the base of the food chain. So we've got things like diatoms and green algae and cyanobacteria and bacteria that are eaten by nanoflagellates, that are eaten by invertebrates, that are eaten by fish, that are eaten by larger fish, for example. Okay, we might ask the question, suppose I were to introduce a new organism, the zebra mussel. Okay, the zebra mussel may change. Let me go back to this slide. Notice the sizes of those different ovals, and let's suppose that the size of the oval relates to its relative abundance. Okay, here we see a fairly balanced food web where there's a lot of links. Okay, where we have relatively even sizes or equity in terms of the different components of the food web. But as soon as we replace the clams with zebra mussels, what we see are changes in the relative abundance of each of these different components. Now, I might want to ask the question, does the addition of the zebra mussel change the food web? In this case, I'm not asking the question, does zebra mussels change the abundance of amphipods? Or does it change the abundance of green algae? Or does it change the abundance of diatoms? If I were asking those questions, that would require that I do 3 separate one-way ANOVAs, right? You know, plus presence or absence of zebra mussels. Does it affect green algae? That could be answered with a one-way ANOVA. Does it affect amphipods? Again, a one-way ANOVA. But I'm interested in the broader question. You know, I'm not interested in do zebra mussels affect green algae? I'm interested in the question, do zebra mussels affect my food web? When I'm looking at the larger pattern, each one of these components of the food web becomes a dependent variable, and I want to answer that question by using a multivariate analysis of variance. Does it affect the food web in aggregate? And to do so, I have to look at multiple different dependent response variables as a function of a single independent factor. Okay, so that's a place where we might want to use multivariate analysis of variance rather than single individual one-off ANOVAs. So another place that you might choose to use a multivariate analysis of variance or analysis of covariance is if you're doing something like community profiles. Here what we're looking at is a DGGE gel of a bacterial community. And if you're not familiar with these, basically the way these gels work is you take an extract of DNA from a microbial community and you PCR amplify a hypervariable region of the 16S subregion of the RNA gene. You then run those out on a gel where there's a gradient from low denaturing to high denaturing down at the end. Okay, so basically you've got a chemical gradient or a thermal gradient along that gel. If you have a sequence of DNA which has a lot of GC pairs, well, the GC pairs don't denature because of the number of hydrogen bonds, and so they migrate pretty well through the gel. If you have something that has a lot of AT pairs, well, they denature more quickly. And basically, you know, you've got this double-stranded molecule that basically forms like unzips, kind of forms like a Y, and becomes stuck or wedged in that gel. So basically what you end up with then is a pattern where each one of these bands represents a different genetic version or variant, and you can think of that as an analog to a different species of bacterium. Okay, so here's within this lane, you can see a number of different bacteria that are present in enough copy number that you end up getting a band. And so we can use this type of a profile to compare this lane with this lane with this lane. Okay, and basically we can say, you know, like this lane has more bands or it has a different number of bands than this lane does. So for example, you see this band here, which is present in this extract. So this might represent one community. That band is absent in this community. Okay. And so we might want to ask questions such as, is this community different than this community? Okay. I'm not really interested in the question, is this band present here and not present there, or is this band more abundant here, a darker band, than it is in this community? But I might want to ask the question on aggregate. Is there a difference between this community and this community? And in that case, what I would look at is each one of those bands, the intensity of each one of those bands becomes a response variable. And I have many different response variables which relate to intensity of each unique band. And I can ask the question, let's suppose that this community comes from a different treatment of some form than this community. Maybe these are growing at higher temperature than these ones are. I can ask the question, does temperature have an effect on community structure? And I would do that by looking at a multivariate analysis of variance, where I'm looking at the response variables, the multiple response variables, each one of those bands representing a dependent variable, comparing this lane with this lane. Okay. I've shown this example also as a multivariate analysis of covariance, and so I should talk a little bit about MANOVA. You can probably already guess intuitively what a MANOVA might be. Okay, in a MANOVA, we might be in analysis of covariance. We had a mixture of discrete and continuous independent variables, but a single dependent variable. In this case, we might have discrete and continuous independent variables, but multiple dependent variables. Okay, so you can imagine that instead of just looking at the effect of temperature, comparing this lane with this lane, suppose instead that these three lanes represented a treatment, a nutrient treatment, for example, or a metabolic treatment. These represent a single treatment at different points in time, and these represent a second treatment at the same points in time. So this maybe is day 0, day 1, day 2, day 3 for a treatment, and this is day 0, day 1, day 2, day 3 for a second treatment. Okay, now we have the type of data set which would require a multivariate analysis of covariance, or MANOVA, where treatment is a discrete variable, a discrete factor, time is a covariate, and each of these bands, the intensity of each of these bands, are my dependent variables. Okay, so that's basically what I wanted to cover in this lecture. So again, when we're talking about multivariate analyses of variance, what we're talking about, or multivariate statistics, what we're talking about are statistical tests where you have now multiple dependent or response variables. And these are basically going to layer on top of a lot of the things we already talked about. You can have multivariate one-way analysis of variance, multivariate two-way analysis of variance, multivariate MANOVA or MANOVA. So a lot of the things we've already seen, just imagine complicating those by adding additional response variables to your analysis. Okay, and that's what we have. So tomorrow we're going to start to look at how to actually do this in practice. So the mathematical underpinnings are a little bit complicated, so we're not going to actually work through and calculate problems in class, but we will go through how to implement MANOVA and repeat measures analysis of variance when we use SAS on Friday. Okay, thank you.